Hello, everyone, and thank you very much for inviting me. It's a pleasure to be here with you. I'm going to go over a little bit about the basic biology of viruses and also extend that to the SARS-CoV-2 as we know it. And I'm not going to spend much on the epidemiology because you have a great timeline in your program there, but I will do a little bit on that, and then we'll discuss a little bit about the origin of the virus and where we stand going forward to mitigate this virus. So where are we right now? Where do we stand? As of yesterday, the numbers I pulled from the CNN COVID tracker said that the United States has 7.1 million cases and almost 205,000 deaths. That's a lot. And in a way, you could say the US is number one, but maybe this is a situation in which we don't want to be number one. In Texas as a whole, again, as of yesterday, over 750,000 cases, almost 16,000 deaths. You can see that epidemiology curve there. To put it in perspective, I thought CNN had a very interesting statistic last Friday about COVID deaths. Our current US COVID deaths are equal to having the 9/11 attacks for 66 days in a row. And since February 6th, our average death rate has been 858 deaths per day. So if the total numbers are not staggering and enough to impress you, perhaps those would make an impression relating to events that have happened in our lives recently. Okay? So a little bit about the basic biology of viruses. Viruses are not cells. Scientists consider them instead to be infectious particles. And this is because they cannot reproduce themselves on their own. They must be inside a host cell. And also, they do not respond to any environmental changes on their own. So viruses are considered to be infectious particles. Whatever the viral genome is made of, whether it's DNA or RNA, that is coded with protein, which is encoded by the viral genome. And some viruses also, including the SARS-CoV-2, have a lipid envelope on the outside. So it's a membrane in which proteins are embedded. And so this is an actual electron micrograph of the SARS-CoV-2 from the NIAID. In order to reproduce, viruses have to enter an appropriate host cell. In the case of the SARS-CoV-2, that typically, at least initially, is a cell in the respiratory tract. Once inside the host cell, the virus uses the host cell's enzymes and the host cell's energy and the host cell's molecules to reproduce itself. So basically, it co-opts the host in order to serve its own reproduction. For the SARS-CoV-2 specifically, I have a cutaway diagram shown on this slide. So on the interior, the genome of the virus is made of RNA, and it's coated with protein. Then this virus also has a lipid envelope surrounding it, within which some viral encoded proteins are embedded. One protein that I specifically want to bring to your attention is the spike glycoprotein, or also called the S protein. This protein is critical for the replication of the virus because it's the protein that the virus uses to attach to the host cell. And that's also an important protein because it's the protein to which vaccinations are now being developed. Okay? So this SARS-CoV-2, like all viruses that infect humans and animals and plants, etc., must enter a host cell in order to replicate. And it basically takes over the host cell's machinery to do that. The SARS-CoV-2 uses that S protein or that spike protein to bind to, typically, initially, a cell in the respiratory tract. And it binds to a protein called ACE2, which is present in high abundance on respiratory tract cells. So the virus attaches to that ACE2 protein using its own S protein, and it's taken into the cell. From there, the viral envelope is opened and the viral RNA is released. Now that viral RNA is just like messenger RNA in any cell, and it can be translated immediately to make the viral proteins. So the RNA is translated. Some of the proteins are inserted into the endoplasmic reticulum and the Golgi apparatus of the cell. And as they are processed, that produces the new envelope for the virus. As the proteins are being translated, the viral genome is also being replicated. So then this whole thing sort of undergoes a self-assembly process such that the viral genome is packaged and surrounded by the new envelope with all the proteins in it, and then it's released from the cell. So once again, these viruses are not living cells. They simply co-opt the host cell's machinery in order to replicate themselves. And in the case of the SARS coronavirus 2, once it's released from a cell in the respiratory tract, it can be released out into the environment through respiratory droplets or aerosols, or it could also go to other places in the body, as we'll see. So as you probably know from the news and all the information that scientists know about this virus, the SARS coronavirus 2 is spread primarily by respiratory droplets. So since its primary site of replication is in the respiratory tract, when we cough, sneeze, speak, etc., those microscopic droplets are released and can be inhaled or drop onto surfaces nearby. And so it's this droplet transmission from coughing, sneezing, these larger droplets, that if inhaled by another susceptible host, could cause infection in that individual. And it's also suspected, many studies have shown, that aerosol transmission can occur as well. So these are the very small microscopic droplets that one could also inhale. If the respiratory droplets fall onto an inanimate surface, such as, the computer desk in front of me, someone could touch that, rub their eye, rub their nose, rub their mouth, etc., and that would be a way of viral transmission as well. Medical professionals have also documented that the virus is shed in urine and feces. That is not considered to be a major mechanism of transmission, but the virus is present in those fluids. Okay. Now, moving on, as far as symptoms go, I'm sure you're aware that the main majority of symptoms are relatively mild respiratory tract symptoms. They can become severe in some individuals. So this can cause fever and chills, in addition to the coughing, sneezing, sore throat, etc. Also, flu-like symptoms that would include muscle aches and joint aches. Some individuals experience nausea, vomiting, and diarrhea. And also it's been noted as an effect of the COVID-19 disease that individuals can lose their sense, or at least have a decreased sense of smell and taste. And unfortunately, in some individuals, this has lasted a long time. But lethal disease is also possible. And that's what makes this COVID-19 so very dangerous and far-reaching in its consequences. So the COVID-19 symptoms, the manifestations of this infection are quite protean. And the Wall Street Journal had a description of it that I thought was quite telling. The Wall Street Journal early in September called it really diabolical. And we know that because not only of its effects on our medical situation, but on our economy and on our own lives. So this virus, while it does normally infect the respiratory tract, it can have a more deadly consequence of causing a pneumonia and inflammation and scarring in the lungs. It also can directly infect cardiovascular tissue, causing inflammation of the heart muscle, can cause blood clots, heart attack, and stroke. It also can infect the kidneys, causing renal damage or renal failure. And it's now been found that it can affect the nervous system, directly infecting the brain and causing encephalitis, which is inflammation of the brain and spinal cord. It can cause seizures, and it can cause a viral meningitis. There have also been patients who had a gastrointestinal infection. And they thought the initial infection was a gastrointestinal site, not the respiratory tract. So these first 5 body systems here, the respiratory system, cardiovascular, etc., those are all sites that can be directly infected by the virus, meaning the virus can replicate in those tissues. As a result of complications in other body systems, though, as an additional effect, COVID-19 can also affect the liver. And then in children, one syndrome that's been in the news lately and discovered because of COVID-19 is this multi-system inflammatory syndrome in children. Basically, it's like a whole body inflammation that causes complications in many different body systems. So this virus can have from benign or asymptomatic disease to something that is very rapidly lethal. And so that really diabolical description is quite appropriate. Where did this virus come from? How did we get it? Why is it affecting us so much? Well, the SARS-CoV-2 is a member of a family of RNA viruses called the Coronaviridae. Okay, so it's known to be a member of this family. That family includes the SARS virus that affected China in 2003, and it also includes the MERS virus, the Middle Eastern Respiratory Syndrome virus. The origin of this virus is hypothesized to be from animals, and that overall the virus sustained several mutations that have allowed it to jump into an intermediate animal host and then to humans. And much like the flu virus can change and slowly mutate over time, so that once in a while we get a very severe flu season, similar thing apparently has happened with this coronavirus. The genome sequence that was released by researchers in early January 2020 shows that the virus is most closely related to coronaviruses that are present in bats. So that's the hypothesized origin of this virus. So it's a zoonotic virus. It is not derived from a laboratory. Scientists who have studied the sequence of this virus across the world do know that it's not lab derived, because to make a laboratory derived virus, they would have to splice together genes from several different sources. And that just is not what appears to be the case with this virus. So it's hypothesized to have originated in bats and then mutated and gone through some intermediate host that then allowed it to sustain further mutation so that it could infect humans. That intermediate host is not conclusively known. For the SARS 2003 virus, it was a civet cat, and that's just a little shadow of what's shown here in this diagram from the Wall Street Journal. But the pangolin is another animal that is proposed to be the intermediate host for this virus. And so it is not known for sure that the pangolin is the intermediate host, but it is known that the first victim of COVID-19 in China did have close contact with wild animal meats at that market in Wuhan, China. So all that being said, it's a bad disease. We're getting some idea of its origin. Where are we going from here? So currently, through the support of Operation Warp Speed, there are 4 vaccines that are in phase 3 trials in the US. 2 of them are viruses themselves, I should say, include viruses themselves, which we hope would generate herd immunity. The other 2 are RNA vaccines. So just a little bit about what we mean by that. The AstraZeneca and the Johnson & Johnson vaccine are a very low virulence adenovirus vector, which means that normal respiratory tract virus has very low potential for causing disease because it's been mutated, but it has the spike protein, that S protein, from the SARS coronavirus 2 inserted in it. So the genome of that adenovirus has been changed to include that the gene for that spike protein. And the idea is that the individual receiving this vaccination will produce that S protein, and their immune system will recognize it as foreign and develop antibodies against it, which would then neutralize a future infection by the SARS coronavirus 2. So the little schematic on the right here just shows this would be the S protein. This would be an antibody produced by the vaccinated individual. It would bind to that S protein and block it. so the coronavirus could not enter that person's cells. The other two vaccines in phase 3 trials are what we call mRNA vaccines. These actually contain the RNA that encodes the spike protein, and it's packaged inside a little lipid particle, what we call a liposome. So it's packaged inside a microscopic lipid particle. When delivered by injection, that particle then fuses with the membrane of a host cell and is taken in. That RNA would then be transcribed to make the S protein. That S protein is produced, the immune system recognizes it and produces antibodies against it. And once again, those antibodies would block the S protein on the real SARS coronavirus 2 and provide immunity. Main difference between these two types of vaccines is that the RNA vaccines do not produce herd immunity. They do not produce herd immunity. So they're only good for that person to whom they've been administered. And of course, we're going to face challenges for distribution of this vaccine or these vaccines when they are approved. Who gets them first? How is the shipping going to happen? Who administers them, etc. So those are things that have to be worked out. And in the meantime, it seems like this disease is not going to leave humanity anytime soon. So we'll need to continue with using masks appropriately and with our social distancing as we've been doing to decrease the spread. Thank you, and I appreciate you inviting me today.